Okay, can I get someone to confirm you can hear me?
I can hear you.
Thank you. All right, before we begin, are there any questions? No questions? Let's proceed then. All right, I think we're in chapter 8. Let me share my screen here. Should be seeing my screen now. Let's get that over here. I don't seem to have that anymore. All right, let's see. View. No, move that around, so I'm a little confused as to where we are. Ah, where's the find? Yep, there it is.
I think we're on chapter 13, not 8.
Chapter 13.
Yeah.
Okay. I'm not finding that either, so maybe that's why I'm not finding it. All right, let's go to chapter 13. Ah, there we go. Well, I don't seem to see a two here or there. Where do I have two of these? Oh, there it is. All right. Yeah, I didn't quite go to the right place. I don't think I've shown the movie on the lysogenic life cycle. I think I did talk about the lysogenic life cycle, but I don't think I showed the movie. So let me see if I can get the link to the movie. It'll start with the lytic life cycle, which I've already showed you.
Is a parasite. Attacks the host cell and takes over it.
Am I sharing my screen? Anyone? Am I sharing the screen?
Yes.
Okay, great.
Yes.
So let's go to the lysogenic life cycle.
Parts are assembled into new virus. Up to 300 new virus parts cell. This process of viral multiplication is called the lytic cycle. Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates and the destructive lytic cycle begins. cell is destroyed, and a new generation of phage released to invade new hosts.
All right. Everything about this movie is perfect, straight on, and all, except for one thing. Yeah, let's go right here. Do a little further.
Yeah.
Right there. Go back a little. And that is it's showing you bacteriophage T4, which bacteriophage T4 does not have the lysogenic life cycle. Other than that, everything about the movie is correct, or the video. And that is it's showing you T4, and T4 only has the lytic life cycle. Any questions about the movie? There are some viruses that only follow the lytic life cycle, and then there are some viruses that are the lysogenic life cycle. And if they are the lysogenic life cycle, then they also can follow the lytic life cycle. Any question about any of that? And what happens, where's my thing right here? What happens is the virus has to decide when it first infects a cell, is it going to follow the lysogenic life cycle or is it going to follow the lytic life cycle? And it follows that life cycle for a time. The lytic life cycle can always stay in that life cycle, but in the lysogenic life cycle, it can leave the lysogenic life cycle and then enter the lytic life cycle. They've studied this now for over 100 years or close to 100 years, and they still don't have a good understanding of why the virus leaves the lysogenic life cycle and then enters the lytic life cycle. They only know one thing that causes the virus to switch from the lysogenic life cycle and then enter the lytic life cycle. And that is if you damage the DNA of this cell, the virus will switch from the lysogenic life cycle to the lytic life cycle. It's like the virus can notice that the DNA of this cell has been damaged and it realizes that this cell may die from the DNA damage, and so it switches to the lytic life cycle. But other than that, they don't have a good feeling for why the virus leaves the lysogenic life cycle and then enters the lytic life cycle. All they know is sometime along the line, every once in a while, the virus leaves the lysogenic life cycle and then enters the lytic life cycle. Any questions about any of that? All right. Some terms you should understand with the lysogenic life cycle. How do I get this blown up? There we go. Is that when the virus infects the cell and the viral DNA incorporates into the chromosomal DNA, this is called a prophage, where the viral DNA is inside the host cell's chromosome. And then there is a repressor protein that's made that shuts down the lytic life cycle. And then the lysogenic life cycle continues. Every once in a while, the repressor protein can stop, and then the lytic life cycle starts. And that can be after a few cell divisions, many cell divisions, or whatever. And let's see, when the lytic life cycle starts, the prophage leaves the host cell chromosome, and we call that excise, and becomes a separate, we call it plasmid, viral plasmid. All right, any questions about the lysogenic life cycle? Move that over. There are some consequences of lysogeny. First, the lysogenic cell, meaning the host cell that has a lysogenic virus in it, will become immune to infection by the same bacteriophage. So this virus cannot infect this cell because it's already infected in a sense. And then another consequence of lysogeny is phage conversion, where the phenotype of this cell gets converted into the phenotype of this cell, meaning this is the DNA of that cell, and now this is the DNA of that cell, meaning it has extra DNA of the virus, and that can change the phenotype of this cell. One example of the phage conversion is the production of a toxin encoded by this virus. And the toxin that I'm referring to is Corynebacterium diphtherium toxin is only produced in the bacteria, Corynebacterium diphtherium, when the bacteria has a lysogenic virus within it. Any questions about any of that? So obviously, if you get infected with Corynebacterium diphtherium, your severity of the disease will be dependent on whether that Corynebacterium diphtherium is itself infected with a lysogenic virus. Because if it is not, then you will not be exposed to the Corynebacterium diphtherium toxin. But if it is, you will be exposed to the Corynebacterium diphtherium toxin. Any questions about any of that? All right, that's enough about talking about bacteriophages. Let's talk a little bit about the multiplication of animal viruses. The basic pattern of viral multiplication follows the same pattern as bacteriophage multiplication, but there are several differences. Like there's differences in attachment. A bacteriophage has to get through the cell wall of a bacteria. And animal viruses don't have a cell wall they need to get through. They only need to get through the cell membrane. And it's much easier getting through the cell membrane than it is through the cell wall and the cell membrane. So penetration is a little different. It can happen in animal viruses by either endocytosis or fusion. The encoding is pretty similar, and that is you can uncoat the virus either by a viral enzyme or a host cell enzyme. Biosynthesis is pretty much the same. You produce the viral nucleic acid and the viral proteins. Maturation is pretty similar too, where the nucleic acid gets together with the capsid to form the new virions inside the cell. And then the release can be the same, meaning we do have animal viruses that follow the lytic life cycle, but they can be different. We have animal viruses that follow the budding life cycle. And all envelope viruses follow the budding life cycle. Any question about any of that? All right, one big difference between bacteriophages and animal viruses is bacteriophages always replicate in the cytoplasm because that's all there is in a prokaryote cell or bacteria cell. Animal viruses, though, can replicate in the cytoplasm or they can replicate in the nucleus. If you have a DNA virus, it most of the time, almost all the time, replicates in the nucleus. And if you have an RNA virus, most of the time it does replicate in the cytoplasm. So that's a rule of thumb. Any questions about any of that? An animal virus, if it's DNA, the rule of thumb is it replicates in the nucleus. And then if you have an RNA virus, it replicates in the cytoplasm. The reason for this is all of the enzymes that the virus needs for DNA replication are in the nucleus of the host cell. And the RNA virus, all of the machinery that the RNA virus needs to replicate, or at least most of the machinery, we'll word it that way, that the RNA virus needs to replicate are in the cytoplasm. Any question about any of that? All right. And then I guess the other big difference is that animal viruses can follow the budding life cycle or the lytic life cycle. And then bacteriophages, they don't have the budding life cycle. They do have the lytic life cycle, but they have the lysogenic life cycle, which only bacteriophages have. The budding life cycle is different than lysogeny. And we will talk about two other life cycles in the animal viruses, but those are the main two. So one difference that animal viruses have compared to bacteriophages is the penetration of the virus into the cell. An animal virus only needs to get through the cell membrane, whereas a bacteriophage needs to get through the cell membrane and the cell wall of the bacteria. Animal viruses get into the cell, meaning penetration, entry into the cell, either by endocytosis or by fusion. Now, some older textbooks, like I think the second edition of the textbook for Clark College, had that wrong, where the textbook said that the virus could enter by pinocytosis, and that's not correct. It's by endocytosis. Does anyone have the older version of the textbook? If you all have the third edition of the textbook or the 11th or 12th or 13th, whatever the edition is of the textbook not made for Clark College, it is correct. Endocytosis is when the virus gets on the cell membrane, and it will attach to the cell membrane, meaning it does have adhesion molecules or ligands on the virus that bind to the cell receptors of the animal cell. And what happens is that the virus will cause the cell membrane to vaginate like this, and then like this, and then this will vaginate even further where this will pinch off, and the cell membrane will reform right here. And then the membrane here, which is the cell membrane, will form around this virus, and that will make a vesicle, bringing the virus inside the cell. And that's endocytosis. Any question about that? Those of you who've had A and P have probably already are familiar with endocytosis, where a what is that? A signal molecule binds to the cell membrane, and then the cell membrane can invaginate, bringing in the message inside the cell. The other way that viruses can penetrate the cell is when the cell membrane of the cell fuses with the envelope of an animal virus. Now, not all animal viruses are enveloped, but those that are enveloped, the envelope can actually fuse with the cell membrane. Let me try and show you that in the show here. So here we have our cell, which is a lipid bilayer that we're going to call the cell membrane. And remember, it has phospholipids with the head on the outside, and then the fatty acid tails on the inside. Trying to draw it here. I'm not going to draw any other than this. Oops, that's not two. Got it in the same spot. There we go. kind of. Making the lipid bilayer. So that's the cell membrane. Well, we have the same lipid bilayer on the virus. Where there's a envelope, which is a lipid bilayer around the virus. And of course, there will be a protein coat inside here. I'm making a rod, which might not be quite correct, but you're getting the idea because I'm not going to try and draw a helix. And then there'll be a nucleic acid for the viral genome. Anyways, where is it? No, that's not it. Yep, I did that wrong. Oh well. Go again there. The make it a little bigger. The trouble with the eraser is I'm erasing very dark. I mean the dark, and so it's light, and I don't know if that's going to work. Let's go here. The envelope can now fuse with the cell membrane there, and then that's there. And now this one fuses with this one here. And that one fuses with this one here. And then the cytoplasm of the cell is connected to the internal content of the virus. And the virus can move through that opening into the cytoplasm. Any question about fusion of the viral envelope with the cell membrane? And the reason why they confuse is because the bilayer of the envelope is made of phospholipids, the same thing as the envelope of the cell membrane. I mean, that's the main molecule in both. And that is showing you probably endocytosis again, but it's also could be fusion. No, that's endocytosis. That's a vesicle being brought into the cell. All right. So animal viruses have two main ways of replicating. That's not entirely true. There is two other life cycles in animal viruses, but the main two are the lytic life cycle, where the virus replicates, destroys the host cell, releasing the newly replicated viruses, very similar to the lytic life cycle of the bacteriophage. And the other way that animal viruses replicate is by the budding life cycle, where the virus replicates its new virion at low numbers, by the way, and then that virus comes up to the cell membrane in a portion of the membrane where the cell membrane has proteins made by the virus, meaning these are some viral proteins. The virus knows that this part of the membrane, like this part of the membrane right there, doesn't have the viral proteins on it. So the virus doesn't come to this region of the membrane, comes to this region of the membrane. And then it pushes on the membrane, forming a bud. And then the virus actually pushes that bud off, ripping off a portion of the cell membrane, which then forms the envelope around the virus, and then the cell membrane reforms, meaning it's broken right here, but then this reforms to that, and that reforms to that, forming the cell membrane. Normally, in the budding life cycle, the virus is shed at low numbers so that the cell can survive the loss of this little portion of the envelope in the bud. So normally in the budding life cycle, the budding virus does not kill the host cell. If the host cell dies, it usually dies from the host immune system recognizing that this cell is infected with the virus, and then the host immune system kills this cell. Any question about any of that? And you can see now why the budding life cycle forms the envelope, and then that envelope is very similar to the membrane of this cell. And that's because that envelope is from a membrane of this cell. It's oftentimes from the cell membrane as shown here, but it could be the nuclear membrane that the virus buds out of to get its envelope, or it could be the membrane of the endoplasmic reticulum that the virus buds out of to form the envelope. It just depends on the species of virus, which envelope it buds out of. All the viruses that I know of actually bud out of the cell membrane, but there are some that bud out of the nuclear membrane, and then a few that even bud out of the membrane of the ER, the endoplasmic reticulum. Any question about any of that? There we're seeing a cell, there's the host cell, with budding viruses budding out of it. And this cell can survive the loss of the membrane there, maybe there, and there. Any question about any of that? And that's actually an electron microscopic image of a cell with budding viruses. These viral proteins are either proteins that are on the viral genome, or they could be glycoproteins that are also encoded by the viral genome. And that's why we call this region of the membrane the viral proteins in the membrane. And that's where the virus will bud out of, because it wants the viral proteins in its envelope. Any question about any of that? All right. Viruses have different genomes and they each require a different way to replicate, like the double-stranded DNA virus has to have a slightly different mechanism for multiplication than the single-strand DNA virus, and very different way of multiplying from the double-strand RNA, although it's similar because double-strand DNA, just the two strands separate, each strand of DNA used as a template to make the newly replicated strand of DNA. And then that's similar to the double-strand RNA, and that is each double-strand DNA RNA will become single-strand, and then one strand of RNA will be used as a template to make the corresponding RNA strand. So each strand of RNA is used as a template to make the newly replicated RNA. And then single-strand RNA is a little different, and that is the replication of a positive single-strand RNA replicates by making a negative strand, and then that's used as a template to make genomic positive strand RNA. And then negative strand RNA replicates by making a positive single-strand RNA, using that as a template to replicate the negative strand genomic RNA. The point is that each virus type needs a slightly different mechanism for multiplying the viral genome. You don't need to memorize this table, but I'm showing you the family of viruses that cause human disease. And there are 7 different viruses, or different families of viruses that cause human disease. So this is the name of the family. We'll talk about a few of them: pox viridae, herpes viridae, adenoviridae. All the others we won't talk about really. And you don't need to know the type of DNA it is or whether it's envelope or naked virus. Although I guess I will mention that the herpes viridae and the pox viridae is envelope. The adenoviridae is naked. I don't think I mentioned that when I talk about the adenoviridae. Capsid symmetry, the size, you don't need to know either of those two. And then just some of the diseases caused by the family. The pox viridae, the diseases we're going to talk about are smallpox and cowpox. Herpes viridae, there are a number of different herpes viruses that cause diseases. Like the simplex virus can cause type 1 herpes simplex or type 2 herpes simplex. Chickenpox, or what's the other one called? The shingles caused by the varicella virus. I don't think we talk about Epstein-Barr virus, but Epstein-Barr virus can cause infectious mononucleosis or Burkitt's lymphoma. And then roseola virus causing the disease, usually in young childhood or infancy, roseola. And then adenoviridae, I'm only going to talk about one thing, and that is adenoviridae is a family of viruses that cause the human cold. And this is actually the third family of viruses that cause the human colds. And it's the least common of the viruses that cause the human colds. The number one is the Picornaviridae, the number one virus that causes the human colds. The number two you actually know of, and it's the Coronaviridae. That family causes human colds, and it's the second most common cause of the human colds. Any question about any of that? All right. DNA animal viruses include the adenovirus and the herpes virus. For DNA viruses, they are generally replicated in the nucleus. So if the virus is a DNA containing virus, it normally replicates in the nucleus. There's only one exception. I don't know if you need to know it. I don't think I quiz you on it. And that is that pox viruses are DNA viruses, but they actually replicate in the cytoplasm. Now, when the DNA genome replicates of a DNA virus for every family but the poxviridae, they're replicating in the nucleus. But the viral proteins are not made in the nucleus of the cell. The viral messenger RNA will be made in the nucleus, and then that viral messenger RNA will go into the cytoplasm to make the viral proteins. And then the capsid proteins are synthesized, and then the capsid proteins need to get together with the replicated viral genome, which is in the nucleus. They need to get together to make the new virions. Any question about any of that? So once a DNA animal virus enters the host cell, one of two things can happen. First, the viral DNA may enter the nucleus, and then the viral DNA can enter the chromosome of that host cell. If the viral DNA enters the chromosome of the host cell, we call it a provirus, a little different term than the bacteriophage, which is called a prophage. This is called a provirus because a phage, that term is referring to a bacteriophage. And then when the provirus forms, that cell will always have the viral DNA incorporated into its chromosome for the lifetime of that host cell. So as long as that host cell is alive, it will have inside of it the viral DNA inside its chromosome. So the provirus remains inside the host DNA permanently. It is never excised. And that provirus can replicate when the host cell DNA replicates. So whenever the host cell goes through DNA replication and then the cell divides, each cell will get a copy of the provirus. Any question about any of that? And then that provirus remains permanently in that host cell for as long as that host cell is alive. The other thing that can happen when a DNA virus enters the cell, the viral DNA can be expressed, making newly replicated genomes of the virus, and then viral messenger RNA can be made, move into the cytoplasm. The viral messenger RNA will be read by the ribosomes to make viral proteins, including the capsid proteins. The capsid proteins will get together with the replicated viral genome to make the new viral progeny, and then the newly replicated virions will be released by the host cell. It depends if the virus is following the lytic life cycle or the budding life cycle, how the viruses will be released. If it's following the lytic life cycle, the virus will lyse the cell, releasing the newly replicated viruses. And if the virus follows the budding life cycle, the viruses will bud out of the host cell. And that's how the virus will get its envelope. Generally, when the virus is following the budding life cycle, the virus is shed at low numbers. And then when the virus is following the lytic life cycle, the virus is shed at large numbers, or like much larger numbers. Any question about any of that? So let's look at the multiplication of a DNA virus in more detail. So the newly replicated virus will bind to the host cell. And there are molecules on the virus called either ligands or adhesions, depending on who your textbook author is, that bind to the host cell receptor, which are specific molecules on the host cell that the virus binds to. And then the virus will penetrate inside the cell, either by forming endocytosis, where the virion will be brought inside the cell by a vesicle, or if the virus has an envelope, the envelope of the virus can fuse to the envelope of the cell membrane, and then the virus will be brought into the cell. The virus, if it's a DNA virus, the DNA will move into the nucleus, and then the viral messenger RNA will be made. The viral messenger RNA will move into the cytoplasm to make the viral proteins, including the capsid proteins. Inside the nucleus, the viral DNA will replicate, making new copies of the viral genome. The copies of the viral genome will get together with the capsid proteins to make the newly made virions, and then the virion will be released either by the lytic life cycle or by the budding life cycle. And then we start the new virion binding to those cell again. Anyways, that's how the multiplication of DNA viruses in greater detail. Any question about any of that? All right, let's talk about the RNA family of viruses. You don't need to memorize this table, but there are 15 RNA families shown here that cause human diseases shown here. We're not going to talk about many of the viruses. We'll talk briefly about the Coronaviridae, Coronaviridae, Orthomyxoviridae, and then where's the other? Did I mention it? Maybe I already mentioned it. Coronaviridae. Um, the Coronaviridae, that's the number one family of viruses that cause human colds. Most of the time, if you get a human cold, it's caused by the members of this family. This family is a very big, very large family and very diverse family. Obviously, it's causing the human colds. Okay, see if I can blow this up a little. Those are the common colds, the rhinoviruses, but it also is a family that causes hepatitis A, and then the family that causes the poliovirus. Each of these viruses are very different from each other. Coronaviridae causes coronavirus diseases like COVID-19. We'll talk a little bit about this family. And this family was actually known to virologists for many, many years before COVID-19 ever came out. This is the second most common cause of the human colds, members of the coronavirus family. Paromyxoviridae is a family of viruses that cause common childhood illnesses like measles, mumps. What is that disease called, causing a respiratory disease? And then you have the seal-like bark. What's that called? Does anyone remember? I'm drawing a blank there on the name of that. Where the child has a seal-like bark. Nobody knows the name of that disease. All right, that's the Paromyxoviridae. Orthomyxoviridae, you probably never heard of that family. That's actually the flu or influenza virus family. And then we'll talk a little bit about Retroviridae. Oops, I meant past it. And those are the retroviruses, which actually AIDS are retroviruses. Any question about the RNA family of viruses? See if I can stop that. Share. Like I said, you don't need to memorize that table. We'll talk about some of the animal viruses. Looking at some of the RNA virus family here, and you can see the viruses are very different. That's an envelope virus. I don't know if that's envelope or not, but very different size. This is a very small virus. This is 100 nanometers, so this is a very large virus. And there's an even larger virus, mouse memory tumor virus. You can see the viruses are very different shapes and sizes. Essentially, an RNA virus follows a similar pattern for the DNA virus in replicating, with obviously some differences because it has an RNA as its genome rather than DNA. Most of the RNA family of viruses replicate in the cytoplasm. There's only one exception, and that's the family retroviruses, meaning the Retroviridae, replicate in the nucleus, the only RNA family that I know of that replicates in the nucleus. When we're talking about RNA family of viruses, if the RNA family of virus is a single strand RNA, you need to state whether it is a positive single strand RNA or a negative single strand RNA. The positive single strand RNA can be read by the ribosome, meaning it'll have AUG on it, And the positive single-strand RNA genome can be read by the ribosomes. The negative strand RNA is the antisense of the positive strand, and it will not have AUG. It will have instead UAC. And the ribosome cannot read the negative strand RNA, single-strand RNA virus. So a DNA virus can easily replicate just by separating the two double strands if it's a double-strand DNA virus, and then using each strand as a template to make the new strand. And then the DNA virus, to make the viral proteins, the messenger RNA will be made from the DNA. The messenger RNA will move into the cytoplasm and be translated into viral protein. So that follows really what's going on in your cells. How viral DNA virus multiplies is very much similar to how the DNA multiplies in your cells. The double-strand DNA virus will simply separate and become a single strand to make the newly replicated strand. And then the DNA will be transcribed into messenger RNA, and then that messenger RNA, the viral messenger RNA, will be moved into the cytoplasm to be translated into the protein. Now, if we have a double-strand RNA virus, it actually replicates very similar, and that's not shown here, but the double-strand RNA separates into single-strand RNA, and each single-strand RNA is used as a template to make the newly replicated RNA. And then you have double-strand RNA, and so you have the genome copy already of the cell. And then I guess the thing is that the RNA can replicate, I guess they replicate the negative strand of the virus to make the viral messenger RNA, which can then be translated into the protein. On the other hand, if you have positive strand, single strand RNA, that strand itself can be read as viral messenger RNA by the ribosome and translated into viral protein. How that RNA strand replicates is it makes a negative strand RNA just by using the positive strand as a template to make the negative strand. And then the negative strand is used as a template to make the positive strand. So that's how we make genomic positive single-strand RNA, and that is you make a negative strand to make the positive strand. Any questions about any of that? Now, if the virus is a negative single-strand RNA genome, it can replicate its genomic RNA by making the syn strand, meaning the positive strand RNA, and then using that as a template to make more of the negative strand genomic RNA. This strand of RNA cannot be read by the ribosome to make protein, but the positive strand RNA can be read by the ribosome to make the viral protein. And so this positive strand is used as a template to make more of the genomic negative strand. And then this positive strand can also be used to make the viral protein. Any questions about what's happening in the different types of replication of the genomic viral genome and how viral protein is made? Why do I have that in there? That's a good question. Let's get rid of that. So once again, looking at a negative strand RNA, the genome will get made by making the positive strand genome and then using that as a template to make more of the genomic negative strand RNA virus. And then the positive strand can be used to make the viral proteins. And then the viral proteins, the capsid proteins, get together with the replicated viral genome to make the new progeny virions. A little different with the positive single-strand RNA virus. It replicates by making a negative strand RNA, then using that negative strand as a template to make more of the genomic positive single-strand RNA. I didn't know that moved. And then that positive single-strand RNA can be used to make the viral proteins. And then the viral proteins, capsid proteins, get together with the replicated viral genome to make the new progeny virions. So the positive single-strand RNA virus, its multiplication cycle is shown here, where the newly replicated virion will bind to the host membrane. There are adhesion molecules or ligands on the virus that bind to the host cell receptors on the cell membrane, and then the virus will come into the cell either by endocytosis or by fusion of the viral envelope, if the virus has an envelope, to the cell membrane. The virus will then be uncoated. This is an RNA virus, so most of the times, except for the family retroviruses, the RNA will be replicated in the cytoplasm. This is positive single-strand RNA, and so we'll make negative strand RNA and then use the negative strand template as a template to make more of the positive single-strand genomic RNA. The positive single-strand genomic RNA, both the replicated and the original, can be read by the ribosomes in the cytoplasm to make the viral proteins, which to get together with the viral genome that's been replicated to make the newly replicated virions, and then those new virions will be released either by lysis of the cell in the lytic life cycle or by budding of the cell in the budding life cycle. Any question about positive single-strand RNA virus multiplication? Oops, did I go past something? All right, let's go to the next one. I thought that was the same one, but it's a different one. The negative antisense strand RNA family of viruses will once again bind to the cell membrane, be brought into the cell. The genome, the negative antisense, will be replicated by making a positive strand RNA. And then the positive strand RNA will be used as a template to make more of the genomic negative strand, single-strand RNA. The negative single-strand RNA cannot be read by the ribosome, but the positive strand can be, and the positive strand will be read by the ribosome to make viral proteins. And then the viral proteins will get together with the replicated genome, the negative antisense strand RNA, or negative single-strand RNA, to make the newly replicated virions, which once again will leave the cell either in the lytic life cycle by lysing the cell, or they will bud out of the cell membrane or another membrane of the cell, but by the budding life cycle. Any question about multiplication of negative antisense strand RNA? viruses. All right, let's talk a little bit about the multiplication of retroviruses, the family Retroviridae. This includes HIV-1 and HIV-2, which cause AIDS in humans. And another lentivirus is actually feline leukemia virus. It causes leukemia in cats, domesticated kitty cats, which weakens the cat's host immune system so that they get a disease very similar to kitty AIDS. Anyone ever hear of feline leukemia virus? Nobody's going to admit it. The retroviruses are unique in that they contain a gene for the enzyme reverse transcriptase. Reverse transcriptase is an RNA-dependent DNA polymerase, which means it can make DNA from an RNA template. And retrovirus actually comes from RE, a reverse, and then TR from transcriptase. So retrovirus is RE, TR comes from reverse transcriptase. There we're looking at a retrovirus, and you can notice the virus is enveloped. It has a viral capsid. See, there's the capsid protein there. And then it actually has 2 copies of positive single-strand RNA. You don't need to know that it has 2 copies, but it is a positive single-strand RNA virus. And inside that virus, there will be a protein called reverse transcriptase. Shown, I think it's that big one there. And attachment, penetration, and coating of the virus, retroviruses, happens as other animal viruses, but biosynthesis stage takes a different life cycle. And that is the positive single-strand RNA gets inside the cell, and the reverse transcriptase will make first single-strand DNA, and then the reverse transcriptase will get on the single-strand DNA to make double-strand DNA. The double-strand DNA will then integrate in the host cell's chromosome, and it can make positive strand RNA, which is the genomic virus, meaning the genomic copy of the viral genome, as well as it can be read by the ribosome to make the proteins of the virus. And then the capsid proteins to get together with the newly replicated genomic RNA to make the newly progeny virions. So let's go through that a little greater detail. Here's our host cell becoming infected with a retrovirus. I'm just going to make an arrow there. The retrovirus RNA gets inside the cell, so let's erase that now. Take me a while. So that is positive strand, single strand RNA. And reverse transcriptase let's go ahead and do that. So, well, I guess we can do this here. I'm going to just go RT for reverse transcriptase. Can make single-strand DNA. And then see if I can select this. Single-strand DNA can be worked on by the reverse transcriptase to make double-strand DNA. Yep. So it does happen in 2 steps. Your textbook only shows it 1 step. And then that double-strand DNA finds the host chromosome and joins with it. I'm not sure how to draw that, so let's just make an X chromosome. And we're going to have the viral genome right here. So this double-strand DNA binds with the host chromosome. Once it does that, it'll permanently be a fixture in this cell, so that this cell always will have the viral genome within its host chromosome. And the trouble is that viral RNA, having a hard time drawing this, can be made. It'll be positive viral messenger RNA. So it'll be really viral messenger RNA can be made, and then that will make viral proteins. Oop. You can put that V for viral. Oop. And then these viral proteins will get together with the newly replicated virion genome, which is actually this right here. That's positive single-strand RNA, to make the newly replicated virions. Let me get that here. And since this is a retrovirus, it will always bud out of the cell because they have an envelope. And then that new virion will bud out of the host cell. Any question about the life cycle of a retrovirus? It has a unique replication cycle where the double-strand DNA enters the host cell chromosome. So it does have a DNA, what do you call it, life cycle, part of the life cycle stage, a double-strand DNA stage in the life cycle of the virus. And that's why retroviruses actually replicate in the nucleus, because the single-strand RNA makes a copy of eventually double-strand DNA, which then goes into the nucleus and combines with the host cell chromosome. Any question about the life cycle of retroviruses? Which is shown right here. So our newly replicated virus binds to the host cell. Oop. Trying to get that out of the way. Can't move it. Oh, there we go. It will probably fuse with the host cell membrane, getting the virus inside the cell. It will make first single-strand DNA and then the retrovirus from the rRNA, and then the, not the rRNA, the positive single-strand RNA. And the single-strand DNA reverse transcriptase will make a double-strand DNA. The double-strand DNA, viral DNA, will integrate into the host cell chromosome, forming a provirus. The provirus will continue for the lifetime of this host cell. And that's the problem with an AIDS patient is that if they get rid of the HIV virus in circulation and they think they're cured, in reality, they could have a cell which has a provirus. And in the future, that provirus could always start replicating more virus. Anyways, the provirus can make viral RNA, which if it's positive single strand can be read by the ribosomes to make the viral proteins, which will get together with the newly replicated viral genome to make the virus, which will always bud out of the cell because it is a retrovirus. Any question about any of that? All right, that's it for the family Retroviridae. Let's talk a little bit about the family coronaviruses, Coronaviridae. They are a single positive strand RNA virus. They have an envelope. And this family of virus, as I mentioned, is actually the second most common cause of the human colds. The only virus that causes more human colds are the family Picornaviridae. Coronaviruses are famous now because they are the family of virus that cause COVID-19, meaning one species of virus, Coronaviridae, causes COVID-19. Another species of coronaviruses cause SARS, a disease similar to COVID-19. And then a third species causes the disease MERS, somewhat similar to COVID-19. MERS, all three of these are diseases that cause severe acute respiratory syndrome. MERS, standing for Middle East Respiratory Syndrome, is the most lethal, with about 36% of people who come down with MERS dying. SARS, about 10% of the patients who come down with SARS die. MERS, fortunately, does not spread person to person. If somebody gets the disease MERS, they likely are spending a lot of time around camels because everyone who's gotten MERS have gotten it from camels, and MERS must be a virus that camels have, and it can cross the species barrier and infect the human. But fortunately, MERS does not spread person to person. COVID-19 can kill. It used to kill at 0.2% to 3% of the patients who got COVID-19 died. This number is actually now half this. So the death rate to COVID-19 is something like around 0.1 to 1.5%. And then COVID-19 spreads more easily than the flu, and one person is likely to infect between 1 and 2.5 other people, which is why COVID-19 continues to circulate in the human population. Any question about any of that? Fortunately, MERS does not spread person to person, and then SARS, which killed about 10% of the people, the health authorities were able to gain control of SARS, and so SARS is no longer spreading in the human population, meaning SARS is extinct, at least in the human population. But SARS actually first spread in 2003, and then it spread one other year, sometime after 2003, I don't remember, where it was spreading in the humans. And fortunately, with SARS, almost everybody, if not everybody who gets SARS, has symptoms. And so they were able to quarantine those individuals and then get control of the disease and keep it from spreading. COVID-19, they tried using quarantine. In fact, if you remember, the United States had quarantine slapped on the country. No one from China could come in unless they went through quarantine for 2 weeks where they were observed to see if they would get COVID-19. And everyone coming into the country, they were scanning them for their temperature and to see if they were sick. And I guess if they were sick, they had to go into quarantine. I'm not sure what happened with her. But obviously, COVID-19, a lot of people can be asymptomatic, and that disease came into this country. And if you don't know, young adults, teenagers, and children, about 50% of the patients are asymptomatic, meaning they have COVID-19, but they have no symptoms. And so they don't even know they're sick. And the trouble is, it won't be deleterious to young people, but then they can spread it to elderly people, such as their grandparents, where it can give a serious disease. If you don't know, COVID-19 is not usually lethal in young people. And about 50% of them have no signs or symptoms that they have the infection. Any question about any of that? All right, COVID-19 is actually closely related to bat coronaviruses. They've sequenced the original coronavirus and they discovered that the coronavirus that was spreading in the human population at the time was a recombination of two bat coronaviruses and a third pangolin coronavirus. The pangolin coronavirus only was 10% of the genome of the COVID-19, but it was an important part. The pangolin coronavirus encoded the spikes on the virus shown here. And it's because of the spikes that COVID-19 can easily spread person to person. They think that COVID-19 spread from bats to probably an intermediate, such as a pangolin. A pangolin is a spiny anteater, by the way, and then spread from the pangolin to a human, and then humans spread it human to human. They think this happened in, I think the city was Wuhan in China, and they think it actually happened in an animal market where in China, they had a whole lot of exotic animals in the animal market. And people would go into the animal market, lots of people, and then buy these exotic animals. And they realized after COVID-19 that twice SARS came out of a animal market in China, and then COVID-19 came out of an animal market in China, and they realized these animal markets having exotic animals was bringing in animal viruses, which then spread to people, and then the people spread it, at least SARS and then COVID-19. So this happened 3 times. And so the Chinese decided this is too risky. We're going to shut down at least most of the animals in the animal market. And so they no longer sell at least pangolins and other exotic animals in the animal market. You can find normal animals like chickens and pigs and probably cows in the animal market and probably ducks, but you can't find exotic animals in the animal markets anymore in China. Any question about any of that? All right. How we know that, oops, got the wrong thing here. COVID-19 was likely a recombination of 3 viruses making COVID-19 was they sequenced the DNA of the viruses. And they've noticed that COVID-19 actually recombined with another virus, and it was actually Omnivon. What was that called? Omicron. There it is. Omicron was a COVID-19 virus recombined with the coronavirus causing the human colds, making omnivirus a new variant of COVID-19. And Omicron was 50% less lethal than the COVID-19 beforehand. And it makes sense it'd be less lethal because it combined with the human coronavirus causing the human colds. And fortunately, all variants of COVID-19 from Omicron have been variants of Omicron. So all of the COVID-19 afterwards have been less lethal than the original COVID-19. It still is lethal, but about 50% as lethal as the original COVID-19 virus. My point being is that we've actually seen COVID-19 recombine with another virus. And so that recombine so far with 4 viruses to make the current version of COVID-19. Any question about any of that? COVID-19 is called COVID-19 because it was a coronavirus, CoV, coronavirus, identified in 2019. That's what COVID-19 stands for. However, that's not a scientific name. COVID-19 does have a scientific name, and its scientific name is Severe Acute Respiratory Syndrome Coronavirus 2, or SARS-CoV 2, which is so similar to SARS, Severe Acute Respiratory Syndrome, but even among scientists, they do not call it Severe Acute Respiratory Syndrome Coronavirus 2. They tend to call it COVID-19. Transmission of COVID-19 is primarily from person to person with droplets from coughs and sneezes within a range of 6 feet. It can spread from further than 6 feet away, and it can spread from indirect contact where somebody sneezes or coughs and the COVID-19 gets on a surface, somebody else touches the surface, and then with their hands touch a mucous membrane in their face, infecting themselves that way. But COVID-19 tends to spread person to person within droplets of 6 feet
I have a question.
and spread other. Yeah.
Oh, sorry. Do you know what RSV is?
Yeah, that's respiratory syncytial virus.
Where would that fall?
I don't remember which family that virus goes into. Do you mind if I look that up right now?
No, you can look at oh, you know what? I think it's pneumonia,
It's a respiratory disease, but I don't know
a type of
which family it falls into. It is a disease that's severe among elderly patients
and
and
children and very young children.
very young children, yes.
Yeah.
See, mostly older adults, but it can hit very young children. Let me go find a better spot. Oh, here it is. Especially infants and older adults. What family is it, though? Not saying.
And RSV is scary because they don't have any like cures or anything really to help. You kind of just have to,
Well, there's no cure for any viral disease.
yeah, run its course.
It's a human orthopneumovirus. What family, though? I don't know this one. Ribovaria. Pneumonoviridae. You know, that must be another virus because I don't have that in the family of viruses that cause human disease. So this must be a new family of viruses that cause human disease. It's a RNA virus, single-strand RNA virus. So there's a 16th family of RNA viruses that cause human disease.
Hmm.
That must be discovered after our textbook said there were only 15 families of RNA viruses that cause human disease. I'm glad I looked it up. Like I said, that's another viral family that causes human diseases. Guess we can shut that down.
Sorry, my phone glitched out right when you said what class it was under.
It's a new class of virus, a new family, Pneumonoviridae. It's not one of the 15 families of RNA viruses that I told you cause human disease. So apparently there's 16 families now that cause human disease because there's a new one. And RSV is the virus causing the human disease.
Oh.
Okay? All right. Our COVID-19 can survive for 3 days on plastic or metal surfaces, but only a day on cardboard or paper. An uninteresting thing of COVID-19 is that it mainly hits elderly individuals. Most viruses, let me see if, excuse me, for some reason I'm burping a lot here. Most viruses, the severity of the disease happens in the young and the elderly. So elderly and the very young, and then actually right here, even young children, the severity of the disease usually drops off in young childhood. And so this is young children right here. And then here, these are easily middle age, even uh, beyond middle age. So I don't know, like age 55. Is that middle age? I think that's beyond middle age. So let's say about age 60. So most viruses follow, trying to get this in here. Young childhood, I don't know. We're just going to go young. And then here, a little past middle age, so let's say around age 60, which this might be a little high, but certainly age 55. Let's go 55 to 60. Meaning in this range from the very young children to age 55 to 60, the virus doesn't do severe disease, only among the elderly and the very young. COVID-19, only the very, very young, like infants, have a severe case of COVID-19. So let's go like that. And then the very, very young, we're talking about probably less than 6 months. See if I've got that there. Trying to do less than, it's hard to do that. And then it's very low to about age 60, and then it starts climbing. And then the elderly, very high risk, much higher than even an infant. And that's COVID-19. Most diseases don't follow COVID-19's severity, where the only very young, the infants are affected, and then the very elderly and the elderly are affected. Most viruses tend to be more like that. But COVID-19 isn't, where the elderly are at very great risk, and then the very young are also at very great risk. And then the young, very young, actually, like 6 months till childhood, teenagers, young adults, even middle-aged, not much risk. You can get sick in middle age, but you're not likely to die. I'm not sure if that's entirely true. Most of the deaths being above age 60 in COVID-19. Any question about any of that? So initially, the actual deaths in the United States were about 1.8%. That number is now about half that because, as I mentioned, the Omicron variant merged with a cold virus that was causing the human cold, and the lethality rate spread about half. Best to stay home if you're sick. Wear a mask if you have to go out so you keep the COVID-19 from spreading. If you need to cough, cough into elbow or tissue. Mortality rate, COVID-19 increased with age. I already mentioned that. Only the very, very young, like infants, it's severe. Ill health of the patient is more likely among men than in women, and blacks and Hispanics more likely than in whites and Asians. They don't know why that's the case, but blacks and Hispanics more likely to die from a COVID-19 infection than whites and Asians. And the same with men. That one's well known that men are more likely to die almost of any infection than women. And it's because they only have one X chromosome. Women have two X chromosomes, which helps them in their defense. Any question about any of that? All right. Enough about COVID-19. Let's talk a little bit about viruses and cancers. Some human cancers are actually caused by a virus, where the person, before they get their cancer, it's well known or correlated that they had a viral infection. Some examples of cancer that are caused by viruses include human papillomavirus causing cervical cancer and skin cancers, and then hepatitis B virus and hepatitis C virus causing liver cancer. In a study, Tang et al. 2013, they found that not all cancers are caused by viruses. For example, breast cancer and glioblastoma, these cancers are not associated with the virus. Obviously, cervical cancer, liver cancer, and skin cancer often are associated with the virus. Any question about any of that? Cancer is just uncontrolled growth of cells. The tumor cells undergo transformation, acquire new properties. Mainly the new properties are they simply continue to grow. Viruses can cause cancer in two main ways, two different ways. You can have a virus integrate into the host cell chromosome that we call a provirus, and that mucks up the DNA. You can mutate a gene if the virus mutates into the gene, meaning the virus inserts itself inside the gene. You can mutate the gene or turn off the expression of that gene, and that can cause cancer if that gene is what do they call that? A tumor suppressor gene. Tumor suppressor genes are genes in our genome that help suppress the formation of tumors. You can also muck up the DNA, and that is the virus can change the expression of the DNA around the region that the provirus inserts into. And so if there's a oncogene nearby where the virus integrates, they can turn on the expression of that oncogene, which can then lead to the formation of a tumor. Now, normally, an oncogene is not in our genome, but a virus can bring it into our genome. And if it's turned off, it will not cause cancer. But another virus can come in and then change the expression of the DNA around where the virus integrated. And that can be a retrovirus or another DNA virus that integrates into the DNA, mucking up the DNA, causing the formation of eventually a cancer. The second way a virus can cause a cancer is with an oncogenic virus. This is a virus that contains a gene that we call an oncogene. So this virus has a gene called an oncogene, which causes the cell that the oncogenic virus infects to replicate, multiply uncontrollably. An oncogene is a normal cell gene involved in cell's growth. But this normal cell gene is mutated, and then the cell will just grow uncontrollably. So an oncogene is a mutated form of a normal cellular gene involved in cell growth that causes the cell to transform by just replicating uncontrollably. And that oncogene is brought into the cell by the oncogenic virus. There are only a few oncogenic viruses, and most of them that I know are actually chicken viruses, but there's some human oncogenic viruses as well. And for some reason, the virus picked up a gene, and then the gene became mutated. And whenever the virus now infects a cell, that oncogene is expressed, causing that cell to multiply. And apparently it's favorable to the virus because there's more cells containing the virus, and so the virus can replicate. Not only by the virus replicating, but the cell replicating. Any question about any of that? All right. I mentioned that there are two main ways that animal viruses replicate. They can replicate by the lytic life cycle or by the budding life cycle. Let me state that there are two additional, meaning there's 4 life cycles that an animal virus can take. There is a latent life cycle, and there is a persistent viral life cycle, persistent viral infection life cycle. A latent viral life cycle is usually a budding virus that while the virus is replicating, it's in the budding life cycle. But the virus can leave the budding life cycle and enter the latent life cycle. While the virus is in the latent life cycle, the virus remains asymptomatic in the host cell. The virus can be in the latent life cycle for long periods of time. And while the virus is latent, meaning in the latent life cycle, it does not cause disease in the patient. And then the virus can leave the latent life cycle and re-enter the budding life cycle, where the virus becomes active, the virus becomes replicating, and the virus causes disease. Two viruses that can be in the latent life cycle or in the budding life cycle, I guess you could say, is the viruses that cause cold sores and the viruses that cause shingles. So when you have a cold sore, herpes simplex virus type 1, the virus is actually in the budding life cycle. The virus is replicating. The virus is causing disease, and that's why we have a cold sore. And as you know, that cold sore can go away. That's because the virus has left the budding life cycle and entered the latent life cycle. And then sometime in the future, that virus can leave the latent life cycle, re-enter the budding life cycle, causing another cold sore. And obviously, the virus is replicating, causing disease. Any questions about cold sores? And then the first time a child is infected with the chickenpox virus, they'll have the disease chickenpox. The virus will then go latent. The virus will no longer cause disease. The child will no longer have symptoms of chickenpox. The virus will go latent, and the virus can be latent for decades. The virus can then come out of latency, enter the budding life cycle, replicating, and cause disease. This disease will be shingles. So all other times, the virus, varicella virus, except for the first time, the virus is in the budding life cycle, the virus will cause shingles. But the very first time the virus is in the budding life cycle, it will cause the signs and symptoms of chickenpox. Any question about any of that? And in between the chickenpox and the shingles, the virus is in the latent life cycle, hanging out, not causing any disease. And this one I happen to know, the virus is really inactive. It's just hanging out in the ganglia cell of your neuron, not causing any disease. The virus isn't replicating. And it can be that way for long periods of time. Usually a child gets chickenpox, and then usually the virus doesn't come out of latency until the child is in old age causing shingles. You can get shingles more than once, and the virus will usually go latent again, but it can come out and enter the budding life cycle, causing shingles again. Any question about any of that? There's usually a trigger for the virus to leave the latent life cycle. The stress of the varicella virus causing chickenpox and shingles is usually old age, where the immune system weakens, and then shingles can come out. Cold sore, the stress is usually the patient comes down with a cold, and then they develop a cold sore. Any question about the latent viral life cycle? There is also a persistent viral life cycle where the virus is replicating, usually at low numbers, and this virus follows similar to the budding life cycle, meaning the virus replicates, but instead of at large numbers like in the lytic life cycle, it's replicating at low numbers in the budding life cycle. And it follows the budding life cycle, except it's a naked virus, but the virus will leave the cell in a vesicle, meaning the virus is a naked virus, meaning no envelope, but it will be incorporated in a vesicle of the cell, and the vesicle of the cell will fuse with the cell membrane, releasing the virus outside the cell, and that's how the virus can shed. And that's called a persistent viral infection. These tend to last a long period of time. The patient usually sheds viruses over long periods of time, and the virus load, at least initially, is very low, and so it doesn't cause harm to even the cell that's shedding the virus, The trouble is this virus can be shed for long periods of time. More and more cells can become infected, and the disease can, well, let's come out here. The disease can be for a long period of time. The severity of the disease on the x-axis, time on the y-axis. Initially, the virus can be shed at very low numbers, but it can pick up over time. And occasionally it can bloom. Let's make that a little longer. Where the virus can then shed at large numbers in a bloom. It's a virus bloom right there. Maybe a bit better if I did it like that. So a persistent viral infection, it can last for a long time, at least initially. Very few viruses are shed. They're shed in a vesicle. So it's a naked virus. Persistent viral infections can occur over a long period of time. The detectable levels of the virus may build up over a long time. The virus may be never cleared by the host immune system, at least in some viral infections. Sometimes the virus can be fatal. I'm sure not many of you know of subacute sclerosing panencephalitis. This is a case of the measles virus entering the persistent viral life cycle, where the virus will be shed over long periods of time, and eventually the virus can bloom and it can be fatal. Another example of a persistent viral infection is HIV in the old days. And this is obviously a budding life cycle virus. But over time, the virus can shed, and then it can bloom, where the virus is shedding large numbers, and then the patient dies. And so obviously the graph is broken there. So at least HIV in the old days, before medication could keep the patient from getting into AIDS, the patient could die. Large number of viruses would be shed at that point, and that would be a persistent viral infection hitting a bloom cycle. Many viruses are tolerated, so you can get a persistent viral infection and the body can tolerate it. Cytomegalovirus, varicella zoster. There are a few patients, see if I can draw that, where the virus hits shingles, the virus is shed, generally at low numbers, and let's do that X square here. And then the virus is shed, usually it reduces at low numbers, and then the virus is just shed for essentially the patient's entire life. My mother had a shingles case where she always had shingles for the last 3 years of her life. So she got a shingles case and then it never went away. She had it for the remainder of her life. And that's usually less than 1 in 1,000 patients. And what she had was a persistent viral infection of shingles. Another example of this, although that one probably goes more like that, is cytomegalovirus, where some people get infected. It's tolerated. They're always shedding virus. There's never a virus bloom, but cytomegalovirus can be shed for the lifetime of the patient. Persistent viral infection. Any question about that? So here we have a normal viral infection called an acute infection, where the virus usually infects the patient, and then shortly thereafter, the virus is shedding virus. So it's an acute infection. And a good example of acute infection is the human cold, where the patient becomes infected. A few days later, the patient is shedding large numbers of viruses, and then the virus goes away. The disease is actually gone, no longer cells infected with the human cold, no longer shedding viruses. That's an acute infection. A latent infection is where the child becomes infected with the varicella virus. They have a first acute infection, getting chickenpox. Very large number of viruses shed when the child has chickenpox. And then the virus goes latent, no virus being shed or replicated, usually for decades. And then the virus can leave the latent life cycle, enter a latent infection where the virus is actually in the budding life cycle, causing shingles. And in most patients, that will last for, what is it? The virus will be in the budding life cycle for 2 or 3 weeks. Shingles will last for maybe about 3 to 6 months, and then the shingles will go away. The latent infection will end. Any question about a latent virus? In between, the virus is latent, the virus is just holding out, not doing anything. And then a persistent viral infection, which starts low levels usually, stays low, can be for many years low, and the trouble is it builds over time, gets worse, and it can bloom. You notice that's broken, where so much virus is being shed that usually the patient dies. It doesn't have to be that way. Like I said, the cytomegalovirus, it can plateau off and then just continue. The patient can tolerate that infection. All right. Any question about latent or acute viral infection, latent viral infection, persistent viral infection? If not, let's talk a little bit about prions. Am I past my time here? I think I am.
Yeah, we are.
All right, let's end this. I'm a little past the time. I don't think we have a lab today. There is no lab today. I will be logging in at 7 o'clock for 15 minutes to answer any questions. After 15 minutes, or the last question is answered, I will log off. Any questions about anything?
I have a quick question. Will you be unlocking the Zoom notes from last week for chapter 8?
You talking about the Zoom notes?
Or your
I haven't
chapter
put
8
it on.
notes?
Yeah, I'll unlock the notes for chapter 8.
Okay, perfect. Thank
Okay.
you.
All right. Any other questions? And thanks for the reminder, by the way. All right. I'm going to end it here.